Well, good afternoon, everyone. Well, thank you for coming to attend to this exceptional seminar. Well, this is my pleasure to introduce Professor Pleuni Penning. So she's from San Francisco State University, and she's been for a few months now here in Montpellier at HISM, and she's going to also stay here in Montpellier for a while. Longer, the better, we hope. So don't hesitate if you want to reach out with her after the talk or also later if you want to talk with her. So Professor Pleuni Pennings, she's an evolutionary biologist. She works, as you can see, on the evolution of drug resistance in HIV, bacteria, Plasmodium. And she wants to determine she wants to understand what determines the rate of evolution in drug resistance. And actually, I discovered she worked on quite a few systems in the past on the evolution of ants, sympatric speciation, adaptation, and selective sweep, with pathogen interaction. Some of our recent work focused on the fitness cost in mutation in virus and the evolution of rats in New York. And she's also very active in the communication and vulgarization of science that she's popularizing with drawings so that she can also attract students to the side of the computational biology, bioinformatics in general. She's from Netherlands. So she did her study at the University of Amsterdam and a PhD in Munich under the supervision of Joachim Hermissen on adaptation and speciation and the modeling of the adaptation and speciation. Until 2009, she was involved or coordinating a graduate program at the Munich University. She did a first postdoc there as well with Susanna Voigt-Sick on ants population genetics. And then, if I'm not mistaken, she moved to the US, to Harvard, to work with John Wickley on, I think it was on HIV and drug resistance. And she continued her work at Stanford under the supervision of Dmitry Petrov to study selective sweep. And in 2014, she took an assistant professor position in San Francisco State University, and she became last year full professor. And so this is my pleasure to introduce and to welcome Pleuni here. Thank you very much.
Thank you so much. Can you hear me on the Zoom? Is it working? It's good? Okay, awesome. Well, thank you, Michael, for that really kind introduction. Thank you all for being here. I've really enjoyed being here today, and in general, I've been really impressed by the scientific community here in Montpellier. And for someone like me who's interested in evolution and ecology and infectious disease and models, it feels like Montpellier is really an amazing place to be. So I feel lucky that I landed here. In the last couple of weeks, I've given the same talk a couple of times about my recent work in E. coli. And I thought today I would talk about something else. So I'm doing something that feels a little risky, which is I'm going to be talking about Plasmodium mostly, even though that is a fairly new topic to me. And I know that there's some people in the room who have lots of experience working on Plasmodium. And so probably there's going to be some, some mistakes and some things that I didn't get right. And I hope you'll tell me afterwards so I can correct it for the next time. How do I go to the next slide? Oh, like this. Okay. So in my last postdoc at Stanford, I was starting to think about what I wanted to do after my postdoc, when I would get my own independent position as an assistant professor. And I worked with a group of graduate students at Stanford on this paper. And in my mind, this was really an idea for what I'm going to do in the next 10 years. Then other things happened this didn't happen, but maybe now that next 10 years can finally start. So this paper, the title of this paper is The Population Genetics of Drug Resistance Evolution in Natural Populations of Viral, Bacterial, and Eukaryotic Pathogens, right? So my idea was, let's do PopGen of all of those systems. And I still think that that is a very, that is sort of what I want to do. No? Okay. And I'm currently writing a book chapter about drug resistance in Plasmodium falciparum. And what I'm interested in for that book chapter and in general is drug resistance, and then the fitness effects of drug resistance, evolution of new resistant strains, changes in allele frequencies, selection coefficients, and mutation rates. And so all of those things, I'm trying to understand how they work together in Plasmodium falciparum. Okay, my very first experience with Plasmodium falciparum was when I was a teen, and I got this amazing opportunity to visit my aunt, who was a nurse in a very small clinic in the eastern part of Zimbabwe. And that was all it was super interesting to be there, of course, as a young person, to visit such a very different country. I'm from the Netherlands originally, so very different from sub-Saharan Africa. But then I got very sick, and well, I felt like I was very sick, but my aunt was not very impressed because she was seeing malaria every day, and she was like, "Oh, let's just check," and yeah, it was malaria. I got some medication, and quite quickly I recovered. But so that was, and I don't remember what medication she gave me, unfortunately. Okay, years later, I was a PhD student in Munich, and I was doing this work on adaptation in populations. And this work was theoretical work, mathematical work. And together with my advisor, we wrote this series of papers about a phenomenon called soft sweeps. And soft sweeps are opposed to hard sweeps, where before these papers, or before the time where we wrote these papers and other people were interested in similar things, in the world of population genetics, people thought that adaptation would usually happen by a single mutation that happens in one individual, and then that mutation over the next generation spreads through the population. Okay, so the idea is suppose that we are all the people here in the room are like a population of some species, and the environment changes, and then whoever is the one who gets the right mutation, the beneficial mutation first, that mutation and the genetic background that mutation lands on, the haplotype that mutation lands on, will spread throughout the population. So, a few generations down the line, at the location where the mutation happened in the genome, everybody will have the haplotype that original mutation landed on. And so then you'll have very low genetic diversity at that portion of the genome. Okay, that was what was thought of, that's called a selective sweep, and it was thought that was the way that adaptation happened. And there were some ideas that may not always be true. And one of the things that happened in the 2000s is that people finally were able to sequence populations, and we didn't find these signatures of selective sweeps the way we expected to find them. Okay, so we did a series of papers, and one of the main things that we discovered is that if the population size is large and if mutation is fairly common, it could easily be that in a population 2 or 3 or 5 different individuals acquire the same or a very similar mutation. And so then if all of the, if these beneficial mutations start increasing in frequency, a few generations down the line, the whole population may carry that beneficial mutation. But now that beneficial mutation sort of lives on different haplotypes. And so you have genetic variation even though the population adapted. Okay, so we suggested, based on math, that under certain circumstances, you could have adaptation to a new environment without loss of genetic diversity. Okay, so this was in like 2005 and 2006. And after I finished this and I was done with my PhD thesis, I was thinking like, okay, I gotta start doing something else because I'm a little tired of this very theoretical, mathematical world where in my mind, it wasn't really relevant to the real world. But then in 2007, this paper came out and it showed me that what I was doing could actually be relevant to the real world. This paper came out from the group of Tim Anderson in Texas. They work on Plasmodium falciparum in Asia, and they described recurrent gene amplification and soft selective sweeps during evolution of multi-drug resistance in malaria parasites. And so they were saying, look, what you're describing in your theoretical papers, it's actually what we see in our data. And so that was very cool. This was one of the first papers that cited our papers. And it was an amazing example of what we called a soft selective sweep. So let me show you a little bit about what they found. So this is a gene amplification event at a gene called MDR1, multi-drug resistance locus 1. If you get multiple copies of the gene, the malaria parasite becomes resistant to a drug called mefloquine. Okay, and so what they found is when they looked at copy number variation along the chromosome, that different samples that they had all had like high copy number variation around this location where the gene is. But where the copy number variation started and ended, or where the amplification portion started and ended, was different in different samples. And so what that shows is that this amplification event must be a different amplification event than this one. Sorry, I'm pointing here, but of course, if you're on Zoom, you cannot see that. Sorry. This amplification event is a different, oh no.
They have on Zoom the video of you. So
oh, they have the video? Okay. Oh, okay, so I can just do this. Okay, thank you. Okay, this amplification event is a different one than this amplification event. Okay, and they in fact identified 15 different ones. Okay, so this type of resistance must have evolved at least 15 times in their sample. And so what they also found is they looked at genetic diversity along the same chromosome. This is, in black is genetic diversity in the susceptible strains, and in white is genetic diversity in the resistant strains. And so diversity in the resistant strains is a little bit lower than in the susceptible strains, but it was not nearly as extreme as you would have expected under a hard selective sweep scenario. So as a comparison here on the right, this is the prediction for like a traditional hard selective sweep where diversity at the locus where the beneficial mutation happened is expected to be like really low. And that's clearly not what they see. So they were excited about our work because they were like, finally, we understand what's happening here. And I was excited because I realized that my work could be relevant. My theoretical work could be relevant for the real world. And around that time, I decided to start working on infectious disease. I didn't pick Plasmodium, I picked HIV at the time, but it became clear to me that population genetics and infectious disease would be a good combination. Okay, not much later, I think 2008 or something like that, I worked on a book chapter for a German book for high school students. I was invited to contribute to that book, and I got to pick my own topic, and I picked drug resistance as a topic because I wanted to learn about that. And so I have this figure in that chapter, which came from another paper originally, but and in this figure, like you see the different malaria drugs or different malaria drugs, and then when resistance was first observed. And so over time, what I realized is that here there's artemisinin or ACT artemisinin combination therapies, and we don't have a little star for drug resistance to them, because at that time, I think there probably was resistance, but I was not aware of it. So I didn't put it in the figure. But not long after that, Tim Anderson's group, again, the people from Texas, came out with a paper showing an extremely soft selective sweep for artemisinin resistance in Plasmodium falciparum in Asia. So what you see here is on the x-axis is time. So this is from 2001 to 2014. And then on the y-axis is the total allele frequency of resistant allele or resistance alleles for artemisinin resistance. And then each different color is a different mutation that confers resistance to artemisinin. And so, for example, at this time point, maybe 40% of the samples carry a resistance mutation, but it's like maybe 15 different mutations that make up the total pool of resistant samples. At the end, in 2014, almost all of the samples are resistant, but actually there's this one resistant type that has taken over most of the population. So all of these different colors are different origins of resistance, but then there's, they have higher fitness than the wild type. That's why they're like increasing in frequency. But then there's one that has higher fitness than all the others. And then that one is taking over. Okay, so now ever since, this is my favorite example of a soft selective sweep. In part because, I don't know, the colors are just so cool. Okay, so let me check how I'm doing in time. Okay, we're good. Okay, so that's two examples of drug resistance in malaria, in Plasmodium falciparum malaria. And so in this case, there's a soft selective sweep that actually reaches nearly 100%. And in this case, the gene amplification that leads to mefloquine resistance, we also see many origins, but actually this one does not reach 100%. And I've tried to find good data, ideally over time or in many different places, to see what levels of, what frequency this kind of gene amplification can reach. And I haven't really found it, but today I've learned a little bit about malaria gen, and I think I can try to figure out more about how common this can become. But as far as I know, this has never reached 100%, whereas this has. And I think that's an important distinction. So I thought it would be interesting for me to organize my thoughts and to organize the data, to think about drug resistance and selective sweeps in Plasmodium as those that have many origins. So those are the soft selective sweeps where resistance has evolved many times. Those that have few origins or even just one origin. Then those that can fix in the population and can reach 100%. And those that stay at low or intermediate frequency. And so the two examples that I gave you, one fits here and one fits here. Both of them had multiple origins, so both of them are soft selective sweeps. But then this one, it fixed, at least in this location, and this one cannot fix. So I'll come back to those later. But I first want to think about this corner. So where there is like few origins, but it can reach a high frequency. And I think the best example for that is chloroquine resistance. So chloroquine resistance has evolved a few times in the world, but not a ton of times. early on, it evolved in Asia, and then that Asian haplotype migrated to Africa, and then it became very common in Africa. And this was during a time where chloroquine was the main drug for malaria in most of the world, but especially in Africa. Okay, so the idea is that, you know, someone who is infected with malaria in Asia that already had the resistance, you know, took a plane to Africa, and then this person was bitten by a mosquito. Maybe that mosquito was already carrying some African Plasmodium, or that mosquito then bit someone else that had African Plasmodium, and somehow the Asian and the African Plasmodium, you know, were able to recombine with each other. And what happened is that that Asian haplotype with the resistance to chloroquine, it reached very high frequency in many different countries in Africa. This is from a recent paper where they looked at this, you know, specific mutation that's part of that haplotype that reached high frequency in the Gambia. Okay, so let me start. So if this haplotype indeed only arrived once from Asia, then this is like the hard sweep scenario where there's only one haplotype that carries a resistance mutation, or actually it's a bunch of different resistance mutations together. And now, you know, over time, if this increases in frequency, what you'll see is that at that position in the genome, you'll see a strong reduction in genetic diversity. Okay, and this is actually what we find. This is data from a paper from 2002. I replotted the data. But so here, the upper line is genetic diversity in the susceptible parasites, and the lower line is genetic diversity in the resistant parasites, in the resistant samples. And you see that at the position where the mutations are, diversity is actually 0. So it's exactly what you would expect in a hard selective sweep. For me, this is still surprising that chloroquine resistance has not evolved multiple times in Africa. You know, there are so many malaria parasites. The continent is so large. Like, why does this not evolve more often? But okay, it didn't. And it led to this, you know, pretty nice sweep signature, I would say. You know, this is like the expectation from a model. Obviously, that doesn't look like this, but it looks a little bit, you know, I think it's still pretty cool. Okay. So what is also super interesting about chloroquine is that many countries, when chloroquine resistance became fixed in these populations, especially in southeastern Africa, countries stopped using chloroquine. And what happened, you know, was better than I think many people expected. That is that resistance quickly went down. So this is, you know, drug resistance, or in this case, it's genotypic drug resistance. So it's the resistance mutations over time in Malawi, and from 1992 until 2000. So that's only 8 years. You had over 80% of the malaria parasites that had the resistance mutation to about 15%. And this didn't just happen in Malawi, it happened in other countries too. I found some very convincing data from Mozambique where the same thing happened more than a decade later, because they stopped using chloroquine later. But again, here from 2006 to 2015, so that's 11 years, resistance goes from nearly 100% to almost 0%. Okay, so if you remove chloroquine, it looks like the chloroquine resistance will die out. Okay, I've just heard today that apparently this is not happening in West Africa. So there must be something else in West Africa. Okay, so when chloroquine is used, and it's probably used a lot because if it's used as first line treatment, you see that resistance increases in frequency. And when chloroquine is not used, resistance declines. Now, that may sound like sort of obvious, but I don't think it's obvious because there's a lot of examples in other systems where drug resistance doesn't behave like this. But this it behaves exactly like what you would expect and what you would hope to see in an evolution textbook. Okay, so what I think that means is the following. So here I'm going to plot. I'm going to plot something, and this line is the fitness of the susceptible allele. Okay, so that's our baseline. And then on the y-axis is the relative fitness of the resistant allele. Okay, and so I think that when chloroquine is used a lot, the resistant allele has higher fitness than the susceptible allele. And when chloroquine is not used, the resistant type has lower fitness than the susceptible allele. And I've put this one a little bit further away from the baseline because it looks like the resistance goes down faster when chloroquine is not used than it goes up when chloroquine is used. But I don't think we have good enough data to be really sure about that. Okay. If you imagine that this is where chloroquine is used a lot, and this is where chloroquine is not used, there must be intermediate levels of chloroquine use, right? Here you could imagine that everybody who needs drugs for malaria gets chloroquine, and here nobody gets it, but here somewhere, maybe half of the people get it, okay? So I imagine that there's a tipping point where the resistant strains and the susceptible strains are equally fit. And as long as we stay on this side, the resistant strain is less fit than the susceptible strain. And I think if resistant levels are really high in your country and you want them to go down, you're better off if you're all the way here, right? You want that resistant strain to be really unfit because otherwise, it will take forever before the resistant strain disappears. But if you're in a situation like in Mozambique, where resistance levels are extremely low, you don't need resistance levels to go further down. You just need to make sure that they don't go up. So as long as you find a level of chloroquine used that makes that the resistant strain is still a little bit less fit than the wild type strain, I think we could be okay. So I think it'd be super interesting to try to find, all the data that is available to see if, this line could be estimated, or this line, the position of this line could be estimated to see how much chloroquine could be used and still not go back to the situation where all the Plasmodium is resistant to chloroquine. And I think that could be useful because chloroquine is cheap and it has few side effects, and it's a good drug and it has a long half-life. And so if we could use it, not for everybody, but even just for a quarter of the patients, I think that could be a benefit. Okay. I'm going to skip this one. Okay. So again, here, these are the ones where resistance evolution happens often. And this is the one where resistance evolution happened a few times. And in the case of Africa with chloroquine, resistance was important only once. Okay. I think this area doesn't exist. So let me try to explain why I think that. So in any situation, I imagine that either the resistant strain is more fit or the susceptible strain is more fit. You can construct models and situations where you know, they can sort of dance around, and sometimes one is more fit and sometimes the other one is more fit. But I think that most of the time, either one is more fit or the other one. And if the resistant strain is more fit, it will increase in frequency until it reaches 100%. That's what you see here, and that's what you see here. If the susceptible strain is more fit, resistance will disappear unless new resistant strains enter the population recurrently. Okay, so in the case of chloroquine, if chloroquine is not used much, and so the resistant strain is less fit than the susceptible strain, you would just not see chloroquine resistance emerge. It's very rare that it evolves. And so if it has low fitness, you should not see it. Of course, there's countries where we see it and we see it have low fitness because chloroquine was used for a long time, so that it increased in frequency. And then when we stopped using it, we saw it disappear. Okay, but if you started from 0 without resistance, and if it had a low fitness, you would just not see it unless it evolved all the time. I think in this situation, we could be looking at a situation where evolution of resistance, like de novo evolution of resistance, is common, but it still has lower fitness than the wild type. So what I imagine for this kind of, for the mefloquine resistance, is that if someone you know, is infected with a malaria parasite and they're taking the drug, maybe that parasite can evolve to become resistant to that drug. And if that parasite has a, you know, so inside my body, because I'm taking the drug, the resistant type has higher fitness than the wild type. But then, you know, once the mosquito comes and it's, you know, it spreads to other people, if the resistant type has lower fitness than the wild type, you know, it may infect a few people, but then it will die out, right? As if it has lower fitness than the other strains around it, it cannot spread. And so what I expect to see in this case is that resistant strains, you know, enter the population because of de novo evolution, but then they die out again. And so you should see turnover of resistant strains, and none of them are able to fix in the population. And so this sort of low frequency resistance that has a lot of recurrent mutation, we can only see it because of the recurrent mutation. If there's no recurrent mutation, we wouldn't see it. That's my prediction. This situation is actually, so for this situation, I don't have a lot of data in Plasmodium at the moment, but actually I think that I know what's going on because this is a situation that I know from HIV and what I think is happening in E. coli as well. Okay, let me check the time. Okay, I have a little bit of time. Okay, so I'm going to switch to HIV. So this mutation, M184V, in the reverse transcriptase gene of HIV, it causes resistance to a drug called Lamivudine. This drug is extremely common. I would, you know, there's millions of people in the world who are taking this drug as part of their HIV treatment in combination with other drugs. The drug was first approved by the FDA in the United States in the early 1990s. And so it's been around for a long time, okay? Resistance to this drug is very common in people on treatment, but in people who are not on treatment is not very common. And here I got data from the Stanford HIV drug resistance database. Over time, I must admit, I don't remember for a second if this is worldwide data or just African data, but I think it probably looks the same. So resistance levels hover around 1%. Okay, so that means that if you have 100 people who are newly infected with HIV, they've never been treated, about 1 in 100 is infected with a strain that has this mutation. Okay, so for a drug that's been around for 35 years, to have it work well in 99 out of 100 people, I think is pretty cool, right? So resistance levels are really very low. But if you look at individual patients, this is data from the 1990s. In this case, the drug was actually used as a monotherapy. So people were only treated with this drug. So that's, we know that in HIV you shouldn't do that, but in the early 1990s, we didn't have a lot of other options. And so if you treat with only this drug, resistance levels inside the patient shoot up really quickly. So you get like very fast evolution of resistance. Each line here is represents one individual, like a person that is infected with HIV. And on the y-axis here, you have the percent of the virus in that person that has the resistance mutation. And so most of these people, within like a week or two, their virus becomes resistant, becomes like nearly 100% resistant. So this resistance is super easy to evolve for the virus. So then why does it only reach 1% at a global level? It's because even though it's fit when you're on that treatment, when you're not on that treatment, it is really not that good. The resistant virus always has a lower viral load than the wild type virus had. Sorry, these are people who are actually still on their treatment. But so even though the resistant virus wins against the wild type virus, the amount of virus in the bodies of these people is lower than, maybe their neighbor who is infected with wild type virus and who is not taking the drug. Okay, so if the viral load is lower, we know that transmission is lower, right? There's a direct link in HIV between viral load and transmission probability. And then the other thing that we know is happening is that you get reverse evolution. So if people are infected with a strain that has this mutation and they're not on treatment, the virus quickly evolves back to the wild type, presumably because the wild type is really that much fitter. So here it is a Kaplan-Meier plot, and it shows the number of people in a cohort that are still carrying the resistance mutation over time. And so what you see is that at about 6 months, you have this very fast drop. And that means that at around 6 months, in most people, the virus evolves back to the wild type. Okay, so between these two things, we understand why this mutation, is not very good at transmitting to other people, and why it has not become a big sort of public health issue, even though it's an issue for the people who are, who are unlucky that the virus evolves resistance in their body. For the people around them, it has never become a huge problem. Okay, and so, sorry. So in this case, we're in this corner where resistance reaches, I would say, a low frequency, but we have many different origins because we know it evolves all the time. Okay, I think in E. coli, we have something very similar happening. Resistance levels in E. coli over time are higher than the case in HIV, but they're also, they stay fairly stable. Here, each line is a different country. And we see here resistance to ciprofloxacin in E. coli over time. And so these lines are never going to reach 100%. So here also, resistance levels, stay fairly low, probably higher than we would like to, but the resistant strains are not winning against the wild type. They're not fixing in the population. And also in E. coli, although the evidence is not nearly as good as in HIV, but we see resistance at many different positions in the phylogenetic tree, suggesting that it must evolve recurrently. And so I think that E. coli also lives here, where resistance evolution happens recurrently, but then the resistant strains cannot win against the wild type. Okay. So to summarize, what I would like to do is I would like to understand drug resistance at the population level in all different systems. Okay, all different systems is a little bit too much, so I'm looking for now at HIV, E. coli, and Plasmodium falciparum. And there are situations where resistance clearly outcompetes the wild type. And there are situations that in a way are a little puzzling because resistance is there, but it's not outcompeting the wild type. And so I think that's because the resistant types are actually not very fit, but they appear recurrently, and that's why they're still there. So with that, I would like to thank my lab and many people at my home institution, ESAM, for hosting me, many people that I've talked to over the years. If you want to find my email address and website and stuff, you can look here. This reminds me that students and professors in the United States are often behind bars, but it also reminds me that she was actually released last week. So if people keep fighting, you know, sometimes good things happen too. She's released, but she did still lose her visa for writing an op-ed. So I don't know. I hope you realize that the situation in the United States is really bad for people, especially for students, especially for foreigners. And we have it still pretty good here in Western Europe. And I'm lucky that I have a European passport. I can be here. Okay, thank you very much.
Okay. If you have question on Zoom, you can maybe write it on the chat and I'll read it out loud, or in here if you have questions. Yes.
Thanks. Can you hear me? Yes. So I was wondering about the difference between the targets of drugs that lead to soft sweeps or hard sweeps. So if there is a class of molecule that is targeted by drugs and most frequently goes towards hard sweep or soft sweeps. And kind of related to this, I was wondering about the hypothesis of recombination between the Asian Plasmodium and the African one. So if that mutation is a hard one, so one that seems to be needed for that molecule to then induce the resistance to the drug, why is there a need of hypothesis of recombination, and this cannot have emerged spontaneously and then fixed because it's the only one? I imagine a recombination model to be needed when there is a mutation that's not maybe really strongly connected to the phenotype, and then it gets fixed in a population, and so you think, okay, maybe it was transmitted. But so
yeah. So okay, so I think so between the soft and the hard sweeps, I think in this case resistance, the haplotypes that cause resistance, they always have multiple mutations. And so that may just make it, you know, more unlikely that it evolves de novo. So for example, the chloroquine resistance, I think it's four different amino acids that are changed in the resistant haplotype compared to the susceptible haplotype. Usually there's only, they use only one of them for like surveillance, but actually it's four different mutations. And so that may just make it unlikely. Maybe it's just statistically unlikely to happen again because you need all of those mutations. Now, why that was possible to happen in Asia, not in Africa, I still don't know, but So that is one thing. And then why we need recombination? I think in that case, you just, I mean, we see evidence for recombination because like the little part that they all share, the resistant ones, is identical to the haplotype you see in Asia, even though like if you go further out, it looks like African, like it looks like the African haplotypes. And so the Asian haplotype must have introgressed into the African population.
Okay. Yeah, so it's not just a specific. And then about the like class of molecules that.
Yeah, I really don't know that. Yeah, I don't understand why there's so many different ways to become resistant to artemisinin. Like all these mutations are in the same gene, but why there's like 20 ways to do that, I'm not sure. Whereas here there's only one way and you need 4 mutations. I honestly have no idea. Maybe other people have ideas, but I don't.
I haven't really kept up with the artemisinin resistance literature for a few years. So what I'm saying, it might be slightly out of date, but there's something profoundly different about what resistant to chloroquine. Resistant to chloroquine is simply a parasite that does not get cleared by chloroquine. So it's a black and white situation. Whereas resistance to artemisinin is a type of resistance that has not been described anywhere else. It's just a slight delay. So the parasite ends up being cleared. It's just that it takes it a little bit longer. So it's a very weird type of resistance. And a lot of those mutations, at least the last time I looked, apart from a couple of them, if I remember correctly, which were really mechanistically associated to a true resistance, a lot of them were just statistical associations. So we knew that some of these mutations had a slightly higher probability of the parasite taking longer to clear, but they're not resistance mutations in the sense that chloroquine resistance is actually either you're resistant or you're not.
Yeah.
So artemisinin is a bit of a weird example.
Yeah. So for the parasite, it's like if it takes a little bit longer to clear, you have a little bit higher probability of still being able to be transmitted to someone else.
Exactly.
Right.
Yeah.
So that's why it helps the parasite, even though for us. So in a way, the artemisinin resistance is not as bad for us as chloroquine resistance. Right, because if the parasite still gets cleared, it means that we still recover from malaria.
Yes, eventually, yes.
Yeah. It's just a little slower.
So this is why you have artemisinin combination therapies, because then another drug comes in and then it actually ends up killing the parasite. So it's you're going to ask a question? And I was just, as a devil's advocate, I don't really see a lot of a difference. The hard and soft sweep, I love it. I think it's very clear and a very elegant example. But in terms of the mechanisms that allow the appearance and spread of mutations, I don't see so much difference between what you, and I know nothing about HIV and E. coli actually, but I don't see that perhaps that much difference. I mean, the mechanisms are, you have drugs, so so they're same for all of them, it seems, right? You have a drug treated population, so you see the drug-resistant mutation increases in frequency because obviously it has an advantages. You get rid of the drug mutation decreases in frequency because it's costly. And then perhaps this drug mutation doesn't completely disappear from the population, it just stays at very low levels, which is probably what happens with chloroquine. And then as soon as you introduce the drug, it goes up again. So, because it's just that when you talked about resistance outcompeting the wild type on the left and not outcompeting the wild type on the right. On the left, you were talking about at the population level, whereas in the right, you were talking about an individual level. Sorry, I might have misunderstood.
Okay, yeah.
The example that you set up on HIV, when they get outcompeted, this is what Andrew Reed's showed that is called competitive exclusion.
Okay, yeah. No, I think I didn't explain. So in this case, like if there's, or like say the chloroquine case, right? Like either resistance goes up or resistance goes down, right? Like,
Yeah.
That's simple.
Yeah.
But in this case, resistance is like straight.
Yes.
And that is not simple, because there's no reason why it would be straight. Either it wins or it loses, but it shouldn't be the
But isn't this explained by the fact that viruses mutate really, really rapidly, so that when they land, you say they acquire resistance in a resistant host, but as soon as they land on a susceptible and an untreated host, they basically lose the resistance because it's very costly. So then it goes back to zero, and then resistance has to evolve again, and this keeps the frequency.
Yes, exactly.
Okay.
Exactly.
Okay.
But that is not the conventional wisdom.
Okay.
So, yeah, but I agree with you.
Okay.
Yeah, it's so, but you need the mutations to appear again and again. Otherwise, there would not be a resistance problem.
And when
right?
a resistant mutation appears, they're often costly. They have to be competing with susceptible, with wild type strains, which are a lot fitter. So this is why they're not able
I
to
agree.
to
I agree
rise in
100%.
frequency.
So you were already convinced, and
Oh, sorry.
and so my story didn't make sense because it already fitted with what you were thinking.
Okay.
Yeah.
Sorry.
Absolutely. Yes.
So I have maybe a naive question, but can you use the type of sweep to predict the genetic architecture of the resistance? Imagine a hard sweep would be a target site.
Yeah.
That binary, you have it, you don't have it, you die. So simple genetic architecture. Soft sweep, many mutation can come in and brings resistance. Might predict complex genetic architecture. So in other words, you could use the type of sweep as a proxy of the genetic architecture.
Yes.
I don't know. It's really a naive question, but I never did the link.
I think, sort of roughly within the same system. Yes, absolutely. Like clearly here we have many different mutations that can do the same thing in the artemisinin case, and here is sort of the opposite. You need like four specific mutations to do the same thing, right? So looking back, it's like, oh, it's obvious that this is going to be hard and this is going to be soft. Yeah. But then in HIV, for example, the mutation rate is so much higher that I don't think it's, because in HIV we have cases where you need exactly this mutation, and still it like happens all the time. So maybe if you move from eukaryote to a virus, it doesn't.
But yeah, that's where you need also the fitness cost. If the cost is very, if the resistance is very costly, then it will disappear quickly and might reappear also later on. In other words, it's just an idea. I don't know why I didn't do the link before, but type of sweeps predict the genetic architecture, and whether it's recurrent or not might also inform on the fitness itself of the. I don't know.
That could be true. Yeah, I think that's an interesting idea to go that way. Yeah.
Merci. Thank you very much for the presentation. So I'm working on host range expansion of viruses. And so I was interested in, do you know if there is any such kind of evidence in host range expansion? Because somehow it's a kind of same mechanism, isn't it? It's like the virus is encounter resistant hosts and has to move and to change to find a way to enter these new hosts. So do you see this kind of soft sweep and hard sweep in host range expansion? Do you have any in the literature? Does it exist or?
I'm not aware of any data, but I'm thinking it sounds like the kind of stuff that Oscar Halacek or Graham Coop are modeling with like soft sweeps in space, or like adaptation in space. It doesn't have to be space in your case, right? It's just a different host that can be in the same space. I have to think about that.
So, hi, thank you for the great talk. So in E. coli, ciprofloxacin resistance is usually due to a chromosomal mutation. For other types of resistance to other antibiotics that are usually used against E. coli, I'm thinking about cephalosporins or carbapenems. They are usually coded by genes on plasmids.
Yeah.
And do you know on which type of models
Yeah.
is right for this type of
Yes.
plasmidic resistance? Is it more like a hard sweep? Like, because you can
Yeah.
easily gain and lose a plasmid, and the plasmid is very costly in terms of fitness. So then the bacteria will lose the plasmids if it's not if you don't take the antibiotic anymore.
Yeah.
So do you know?
Yeah.
Do you have?
Yeah. So that's an interesting question. So I think it depends on whether your focus is on the plasmid or your focus is on the bacterial strain that has or does not have the plasmid. So if you imagine the bacterial strain that acquires the plasmid, and then the strain and the plasmid together, you know, they can spread to other people. Then in that case, I think it looks like this actually, where like strains acquire plasmids, but then, as you say, there's a fitness cost, and so they lose it again, and so we continuously see these resistances like at some level, but they never take over the entire population. You know, probably because there's a fitness cost. That's why like resistance levels are, you know, fairly stable and fairly low most of the time, though not everywhere. You know, I think it could also be interesting to sort of look at things from the plasmid perspective, and you know, the plasmid may always have that resistance. So that may be a different situation, but I think from the if the bacterium is the unit that you're interested in, I think it looks like this.
Okay.
Too.
Thanks.
Thank you.
Hi, thank you very much for your talk. I'm wondering if you know the reason why the resistance to chloroquine and to artemisinin appears in Southeastern Asia.
Why that's like a hotspot for resistance?
It seems to be a hotspot for
Yeah.
resistance.
Yeah. So I really don't know. I can only, you know, repeat a little bit what I've read. And one of the things that I've heard is that there's a lot of places. Okay, so there's places, for example, in Thailand where malaria is very common, and there's places where malaria is not common. And then if people move from one place to another, so in some of the places where malaria is common, there's work, you know, there's mines or there's like forests or like there's like work. And so you have migrants, people who move. And so if you have adult people who have no prior exposure to malaria and they go to a malaria endemic location, apparently you get like, you know, a lot of infections, and that can presumably, I don't know exactly how it will work, but create a situation where you have a lot of, you know, where there's opportunity for evolution. And then I've also heard that in some of these places, they use a lot of malaria drugs. And I've recently read that in the case of chloroquine, they used to put chloroquine in the salt so that everybody is treated all the time. Okay. So, you know, that could maybe explain the chloroquine situation. So, but I don't know, because I know that like for artemisinin or others, like I don't think anyone puts that in the salt. So, so I've heard that that may be the reason that you have like these people who live nearby who are not normally exposed to malaria. Whereas in Africa, or at least in Southeastern Africa, it's like everybody is always exposed to malaria. So you get it as a kid, and that may lead to different situation. I would love to understand that more, but this is what I've understood.
Okay, thank you.
Merci. I mean, just to say what you just said, indeed, the situation in Asia is very different because virtually every case of malaria in Asia will get treated. In Africa, it's the other way around. Most cases don't get treated. So the pressure is very different for the parasites. One thing we know that there is no difference of is the mutation rate. So the mutation rate in African falciparum is the same as the mutation rate in Asian falciparum, at least in vitro, because that's where you can measure it. But what has always surprised me is the mutation rate is actually relatively high. Or more specifically, in a falciparum infection, you have millions or billions of parasites. So if you do the math, and I've done it before, virtually every single nucleotide of your genome is mutated in a normal falciparum infection. So why don't you get chloroquine resistant after every single time you take a tablet? I don't really know. And probably it's other region of the genome that's the genetic background, sorry, could explain it. So it's not, of course, the mutation K7060 is critical, but maybe the rest of the genome is also important to a lesser extent.
It's also because I think chloroquine resistant is one of the costliest one in terms of fitness for the parasite, apparently. And also, because it's very costly, it has a lot of like metabolic costs for the parasite. So this parasite, which if it appears in a host, is going to appear in a very low frequency. So there's already stochastic processes that might make it disappear, but it's also going to find itself in competition with wild type parasites that they don't have. So I think making spontaneous chloroquine resistant appear and maintain is perhaps not that easy actually.
Yeah.
It's a particularly costly one.
Yeah. But I do agree that, you know, there's definitely a question there, not just in malaria, but in many other populations where we, you know, if we multiply the number of individuals with the mutation rate, things must be evolving all the time, and we don't see it. For ciprofloxacin, you know, people have tried to find out, like, okay, if you have E. coli in your gut, you take ciprofloxacin for a couple of days, and most of the time ciprofloxacin resistance is encoded by 1 or 2 mutations, like single nucleotide SNPs. You know, you would think that the E. coli in your gut, there should be enough of them to become resistant to this ciprofloxacin easily. And yet most of the time we don't see it happen. And so I think, you know, that question between, like, you know, the math of the mutations and the individuals and like the actual adaptation or evolution that we see, it doesn't seem to add up. And I don't think, you know, I don't think we really know that. You know, backgrounds probably matter, but at the same time, we know that there's a lot of recombination, and like that one, you know, allele at the PFCRT gene, it did spread through Africa without necessarily being linked to many other things. So I would say that's still an open question. But one thing that I was just thinking is, you know, I had this drawing here about like having a lot of chloroquine treatment and no chloroquine treatment. And so basically what you're saying is that maybe, you know, maybe this is like the situation in Malawi or something like that, but maybe in Thailand, we're like all the way over here, and like the benefit of having chloroquine resistance is even higher. I mean, then you still need to be able to combine all those mutations, you know, to get that allele, but, you know, so maybe, you know, maybe this should be expanded.
There is a question from the chat
okay.
from Frédéric Simard. Very nice talk, thank you. For falciparum, what do you know and how do you account for neutral population structure? Might be different in Asia versus Africa. See multiplicity of infection in humans and vectors. And there's another question. How do you account for the fact that the parasite develops in 2 distinct environments, mosquito and human, where selective pressures might be very different and the same mutation might have different effects on parasites biology?
Yeah, okay. Well, those are difficult questions. So the one about like population structure, I really don't know much about that. What is sort of surprising to me is that, how like in Malawi, resistance disappeared between 1992 and 2000. And in Mozambique, which is, not far away, like the same thing happened, but like 15 years later. And so it seems that, at least these different countries, and like we have data for Zambia and Kenya, and like these countries, even though they are not too far away from each other, they seem to behave sort of independently. And so that suggests to me that there must be, migration between those countries must be somewhat limited. Otherwise, like things would just all look the same. But how that structure influences, evolution of resistance, I don't know. And then the second question was about the difference between the mosquito and the human. And I think, and I know that Anna has worked on that, so I have to be careful here. Okay, so first of all, I think the resistance mutations that we actually see and observe, they must be, they must be able to, live in the human and in the mosquito. Otherwise, we wouldn't see them, or we would see them very rarely. And actually, resistance to Malarone or to Atovacone, which is part of Malarone, we know that it appears sometimes in people who are treated with that drug. But then people have showed that that mutation that causes resistance is lethal in a mosquito. And so in that case, that mutation will never spread widely because it cannot move from one person to another. So that mutation is rare. And so, we usually only see it in travelers who come back and who, whose malaria doesn't clear when we expect it to. And so that mutation will never become a big problem because it doesn't survive in a mosquito. So in other words, once we see it as a problem, it probably means that it's okay in a mosquito. Yeah.
Thank you. Are there any other questions in the chat, or? I think there was only that question, or those two questions in the chat.
Thank you for the talk. In fact, I would be very interested to discuss with you about Mycobacterium tuberculosis. I don't know if you know this bacteria, because it is a very incredible bacteria, because the positive selection is really specifically on genes and on specific region of the genes for drug-resistant genes, in fact. And we have a lot of epistatic effect between the drug-resistant genes, and also the emergence, when you have emergence of drug-resistant mutation, for example, you have also in parallel to decrease the cost as the emergence of compensatory mutation. So all this aspect, I think that in the different examples that you took, in fact, you did not have this problem, for example, for chloroquine resistance or the other one. But I think that it can change also the model.
Yeah. I think Mycobacterium tuberculosis is super interesting. And actually, I had a master's student who worked on that last year. I find it complicated.
Yes.
And what is happening in different countries is really different. Like in some countries, there's like resistance has spread, has become a huge public health problem. And so I would love to discuss
Yes,
because I find it a little overwhelming, but certainly very interesting.
and
It's a-
also depending on the resistant genes, you don't have the same model, I think, because for example, for PNCA genes, we have a low frequency of each mutation, for example, but for the RPOB genes, we have a frequency 80% of one mutation. So I think that's really interesting
would be
because you
super
have all
interesting.
all the
options.
Yes, all the options, and also it is chromosomic, only chromosomic mutation.
Yeah,
and
but it
is
doesn't recombine, right?
exactly, it's a clonal
Yeah.
species. You don't have plasmid, you don't have recombination. So I think it would be very interesting to see, to explain, and also to see for some, maybe because now we are using multiple antibiotics for this in same time for this species, but maybe in fact if we look for the model, maybe we can say, okay, for this treatment or for these antibiotics, we need to decrease the use of this antibiotic specifically to avoid the effect on the other resistant genes. So I think it will be very, and I think that not a lot of people study this bacteria with because it's complicated. I think in terms of modeling, and so it will be very interesting to.
Yeah, I would love to chat about that.
Yeah.
Thank you. Any other questions, or are we done? Thank you very much for your presentation then.
Thank you.